Okay, dear students, myself Dr. Navneet Parekh, instructor of course APS 221. The title of this course is Agricultural Microbiology. Uh, Before COVID-19 situation, we have already studied some of the topics. Now I'm proceeding with a new topic, that is internal structure of prokaryotic organisms in special reference to the bacteria. Uh, Today we'll discuss some internal structures. So beneath of the external structure, there are many internal structures that exist in the cell, like cell wall, then the periplasmic membrane, then the cytoplasm, then nucleic material which is embedded in the cytoplasm, then ribosomes, and etc. etc. I mean, there are many. So external to the cytoplasmic membrane, there is a structure that is known as a cell wall. So today we will discuss about the cell wall of the bacterial cell. This cell wall is a rigid structure and give shape to the cell, and it also prevent the cell to burst due to the adverse external medium. Sometimes or many times you can say these bacteria they remains in the hypertonic solution. Hypertonic solution is a solution in which the lower osmotic pressure uh, then exists in the bacterial cell. So what actually happens with this, that all the material which is outside to the cell tries to move into the cell, and eventually the cell can burst, and this cell wall prevents with this. To study the cell wall structure, we have to prepare, or we need to prepare the slide of the cell wall. So definitely in that case we will rupture the cell wall by maintaining or exposing it with the extreme pressure, and then immediately that pressure is being released, and because of that the cell bursts or wall gets turned off. So the parts of that cell wall then embedded in the medium, and that medium can be used to prepare the slide. There are other method by which you can take the parts of the cell wall by putting ultrasonic treatment. So with this, you can prepare the cell wall slide. In general, it will see the cell wall, the size or thickness of the cell wall is more in case of gram positive than the gram negative. And the same situation also remains, situation remains similar in case of gram negative and gram positive archaebacteria. The gram positive archaebacteria uh, then thicker than the gram negative archaebacteria. And bacterial cell wall are usually essential for the bacterial growth and division. As far as the composition of the cell wall is concerned, the main shape determining part of the cell wall is known as a peptidoglycan. Sometimes it also known as a murine. It has 6 different polymers or components, you can say. They are listed like N-acetylglucosamine, N-acetylviramic acid, L-alanine, D-alanine, D-glutamate, and L-amino acids. N-acetylglucosamine is also known as NAG, then NAM. So this way they are also known as with their short forms. Uh, these walls of prokaryote archaebacteria possess or sometimes do not contain the peptidoglycan material. So this type of the bacteria, those who do not contain peptidoglycan material, they contain or they are composed of the protein like glycoproteins or polysaccharides. Cell wall of methanobacterium, for example, the cell wall of the methanobacterium composed of pseudomurin. So other than the peptidoglycan material, there are some other material through which the cell wall is being composed of. Then the differences, we will discuss the differences of the gram positive and gram negative cell walls. These are the main differences you can find in gram positive bacterial cell wall, which is thick, have much greater amount of peptidoglycan than the gram negative bacteria, and it also contains teichoic acid, polymers of the glycerol, ribitol, and phosphate groups are also there. Linked, sorry, this is not linked by the phosphate groups. Then the walls of the most gram-positive bacteria contains very little lipids in it. Some mycobacterium and cyanobacterium, they are rich in this, and though they are gram-positive, they contain lipids. But other substances may occur in addition to peptide glycan, like Streptococcus pyogenes. This is one of the bacteria which is gram-positive. It contains polysaccharides, and similarly, in this case, it contains polymers of the ribitol phosphate or glycerol phosphates. In case of gram-negative bacteria whose thickness is 145 angstrom, uh, is thinner than the gram-positive bacteria. Most important difference presents is the outer membrane. In this case, the outer membrane will be there. In gram-positive bacteria, outer membrane is absent. Because of the outer membrane, gram-positive bacteria are rich in lipids. Okay, so outer membrane serves as a impermeable and which gives more resistance to the antibiotic or provides resistance against the antibiotic. Then outer membrane also serves as a barrier to various external chemicals and enzymes to prevent, this is not to prevent, to prevent from the damages. Outer membrane enclosing the periplasmic space which contains enzyme involved in nutrient acquisition and toxin. So this outer membrane also works or helps in the nutrient acquisition. This is the main difference you can see here. This is the gram-negative cell wall and this is the gram-positive cell wall. If you want to see, you can see here that in gram-negative cell wall there is a less peptidoglycan material, or rather outer membrane, the size or the presence of the outer membrane is there. In this case, in the gram-positive cell wall, outer membrane is not there, but teichoic acid is there, and this one is the peptidoglycan material, and below this, from this periplasm, below this, both these internal structure present in case of gram-negative as well as the gram-positive. So these differences are there. So here this outer membrane is in addition in comparison to this. Here the teichoic acid material is there, the lipid is, this is rich in lipid, so lipid material is there. Then in summary, you can see, you can read in the gram-positive cell wall, the gram-positive cell wall is characterized by the presence of the peptide glycan. This all, yeah, we have already studied, and because of the lipoteichoic acid and lipids, different lipids that is there, which is covalent dealing with the other lipids. Teichoic acids give the gram-positive cell wall as an overall negative charge due to the presence of phosphodiester's bonds, okay, in between or between teichoic acid and monomers. So this is the summary of the gram-positive cell wall, whereas the gram-negative cell wall contains thin peptidoglycan material, but it contains outer membrane, and in addition to the peptidoglycan layer, a small, very thin layer is there, and also contains the outer membrane. As a lipopolysaccharides are highly charged, the gram-negative cell wall has overall negative charge on that layer. Then this, you can see, is often unique to specific bacterial strains and is responsible for many of the antigenic properties in this strain. So it provides protection. This outer membrane provides protection against the antigen material or antigenic material. And below that cell wall, the other structure, that is the cytoplasmic membrane. This is the cytoplasmic membrane beneath to the cell wall. So in that previous structure, this was there, which I have prepared by my own. So you can see here, some are the integral proteins are there, some are the peripheral proteins are there. These are the phospholipids. So this is the schematic interpretation of the cytoplasmic membrane. What it does, integral protein, it is tangentially helping in the cytoplasmic membrane, which holds or attach with the peripheral protein, or it holds the cell wall and gives the density to the whole cell. The lipid matrix of the membrane has fluiding, allowing the allowing the completes to move around the, literally means the whatsoever material is inside the cell, it helps to it helps the cytoplasmic material in streaming movement. So that is there. Then the other thing which is there in the cell, uh, internal part of the cell, that is protoplast. You all know the protoplast is that portion of the bacterial cell consisting of the cytoplasmic membrane and the cell material bonded to it. Protoplasm can be prepared from gram-negative, as means protoplast can be prepared from gram-negative bacteria as well as the gram-positive cell. Here, gram-positive cell. Sorry. Here, the cytoplasm. Cytoplasm is again uh, is a material which is there is a fluid material which is there in the cell. It has three major area or components, like cytoplasmic area, then chromatin area, and then fluid protein uh, with dissolved substances. So that material is fluid material which remains in the cell. It is a granular in appearance and rich in macromolecular RNA protein bodies known as ribosomes, on which the proteins responsibility to synthesize the proteins. Chromatinic area, which is rich in the DNA. So this is there. This is the cytoplasm. The ribosomes are there. You all know about the ribosomes. It contains two different types of the ribosomes. 30 means two subunits are there, 50 and 30 S, and opposite to in eukaryotic cells, it contains 60 and 40 S. So in prokaryotic cells, you can find two subunits, 50 and 30 S ribosomes. These ribosomes are not bounded with the membrane and they remains embedded in the cell, and these ribosomes are having responsibility to form or to provide the message or to help in the formation of proteins ultimately. So these ribosomes are again the important part of the cell. The other thing is the nucleic material. The bacterial cell that contains nucleic material that is not membrane bounded, so that remains embedded in the cytoplasm, but some nebulous material is there. So in the center of the cell, nuclear material uh, remains these and regards as a nuclear and DNA confined area. So in that nebulous structure, that whole structure where the DNA material is there or nuclear material is there, that is known as a nebulous structure. And as we designed as a nucleotide chromatin body, nuclear equivalent or bacterial chromosomes are there. So as such, the true nucleus is not there, but that all material that remains present, and this nuclear material is having a tendency to divide, or it divides into different parts. These were the internal structure of the cell wall. Then there are some bacterial cells, they form these spores. This is not the internal one. I'm not talking now the internal structure. Now I'm talking about the different types of the bacterial cells. They have different properties. Like some bacterial cells, they have property to form the spores or cysts. So certain species they form pseudospores. Certain species they form the cysts. These spores and cysts are being formed to protect the cell from the outer external adverse or stress conditions. Whenever these spores comes under the appropriate conditions, they again starts germinating. So these are of two types, endospores and exospores. Endospores are having thick walled bodies, whereas the exospores, these are being formed external to the vegetative cell by budding or at one end of the cell. So some different types of the endospores and exospores are there. Then the cell metabolism. And now I'm starting with a new topic, that is cell metabolism. Cell metabolism is again an important topic, which contains how cells they take their nutrition. So every organism which is present on this earth, they require three things. One is the reducing power, then another is the carbon dioxide, and then energy. So these three things they acquire from the nature. Actually, in our course, we have these two terms, phototropy and chemotropy. So, first of all, to understand these things, we should know about the phototrophs and chemotrophs. The phototrophs, they obtain energy and reducing power from the radiation, and carbon from the carbon dioxide. So, all these three they acquire from the nature: energy, reducing power from the radiation, energy from the light, and then this carbon from the carbon dioxide. The whole process is known as a phototrophy. And whereas the chemotrophs, the organisms which obtain their energy by oxidizing electron donors, are known as a chemotrophs. And their carbon sources can be either inorganic carbon or organic carbon. To better understand this, I'll provide you some links, and you kindly go through with that or those links, which will help you to understand these phototrophs and chemotrophs. Here again, I have tried to prepare their differences. Here the phototrophs in blue color chart, you can know about the phototrophs, whereas the green color chart, you can know about the chemotrophs. Just read and then go through with that links which I will provide you in the last of the slide. Here are some again differences. In some of the differences I just want to explain. Here metabolic requirements are this: energy, reducing power, and the carbon. The sources, in case of energy, the sources, if they are deriving from light and mode of nutrition, then it will be known as a phototropy. And representing microorganisms, the examples are here. The same way you can see, and you just remember them for better understanding of the bacterial nutrition. And these are the two links which you can see, and go through with these links. They will explain you what is phototropy and what is chemotropy. Thank you very much. I'll come with some new topic within 1 or 2 days. Thank you very much. Bye bye. Thank you.